Okay, so today I'm going to discuss with you about the enzyme called topoisomerase. Now, this is the you can say it is a main important enzyme which overcomes the supercoiling of the DNA. Now, what is supercoiling? Actually, when the helicase enzyme unwinds the two strands, there will be a tension force created, and due to which over twisting will be observed ahead of the replication fork. To overcome that over twisting, which is also called as positive supercoiling, we have to minimize the tension by cleaving, by cutting one of the strands or both of the strands with the help of topoisomerase, and then overturn them, we underturn them, okay, to make them normal coiling condition, and then we will reseal them. So that is done by actually topoisomerase. So topoisomerase are the enzymes that participate in overwinding and underwinding of the DNA. Overwinding means if there is very loosely coiled, okay, suppose like this, if there is suppose very loosely coiled, okay, so you can see that not too much of winding has taken place. The DNA double helical nature has been um, loosely coiled. So the topoisomerase will try to um, make that overwinding. So it will try to make that overwinding to make them normal DNA look like this, and it also do underwinding also. Underwinding generally occurs when supercoiling of DNA occurs ahead of the replication fork. Due to unwinding of the two DNA strands, what happen when helicase move along with the direction of the replication fork, then what happen ahead of the replication fork, too much of coiling occurs and not a type of knot occurs that leads to overwinding of DNA. To minimize that problem, topoisomerase carry out that function by cleaving one of the strands or both of the strands, then minimize that force, minimize that knot conditions, minimize that overwinding conditions. It undergoes underwinding and then again reseal the strand with the help of their ligase activity. So this winding problem of DNA arise due to the intertwined nature of its double helical structure. Now, during DNA replication, and also it is observed during transcription, when you will study that when DNA is being used as a template for generation of RNA strand, during that time DNA has to unwind, and during unwinding of the DNA as done by the helicase, and in case of transcription it is done by the sigma factor, you will see that the DNA becomes overwind, or you can say overowned ahead of the replication fork. So if this is the replication fork, sorry, okay. So if this is your replication fork, I have to show you by helical diagram, okay. If this is the replication fork unwinding by the helicase, so as it is moving from 5 prime to 3 prime direction, as it is moving from 5 prime to 3 prime direction, what happened, helicase is moving in this direction ahead of this replication fork. That means in this part overwinding of DNA occurs, twisting, more twisting of the DNA leads to knot formation, okay. So to overcome this problem, DNA topoisomerase come into action, okay. So DNA becomes overowned or overwind ahead of the replication fork, and if that overwinding of DNA is left unabated, means if it is not solved, then this overwinding of the DNA, the torsion, okay, will eventually stop the ability of DNA or RNA polymerase involved in the process to continue down the DNA strand. So helicase is unwinding, and due to which what happen ahead of the replication fork, overwinding of the DNA occurs. Now, if you don't solve this overwinding of DNA, what will happen later on when DNA polymerase or RNA polymerase in case of transcription, RNA polymerase in case of DNA polymerase 3 in case of replication, they will actually they will eventually stop. Okay, no more unwinding of the DNA occurs because too much of torsion, too much of force is created due to knot formation. Now, in order to prevent and correct these types of winding problems caused by the double helix, topoisomerase bind to the DNA and they cut, or you can say cleave, the phosphate backbone of either one of the strand or both of the DNA strands. And this break, the break that is caused not is permanent break, it is an intermediate break. After breaking that strand, it allows the DNA to be untangled. Untangle means to uncoil, that supercoiling. Okay, or you can say the DNA becomes unwounded. At the end of the process, the DNA backbone is resealed again. Simple thing, that what does topoisomerase do? In that supercoiling region of the DNA, topoisomerase first bind to the DNA and then it cut the phosphate backbone in that supercoiling region. Depends on what type of topoisomerase we are using, it will either cut one of the strands or both of the DNA strands. And due to cutting of these DNA strands, it allows the DNA to uncoil, to untangle. Okay, so that the supercoiling may be relaxed into its normal coiling form. And again, at the end of the process, the DNA backbone is resealed again by the same topoisomerase enzyme. Now, topoisomerase, as I have said, depend on what type of break it is occurring. It is creating break in one of the strand or both of the strand. Topoisomerase are of two types. One is your type 2 topoisomerase, which make transient double stranded breaks. Transient means temporary double stranded breaks in the DNA, through which they pass a segment of uncut DNA, duplex DNA, before resealing the break. So, what does type 2 topoisomerase do? They make temporary double stranded breaks so that they can pass a segment of uncut duplex DNA before resealing that break. Type 2 topoisomerase, they require energy of ATP hydrolysis for their action. Now, they do not require energy for cutting those strands. Actually, they require energy for changing their for conformational change actually in the structure of topoisomerase 2. Later on you will see. Okay, both type 1 and type 2, they type 1 obviously they don't need energy, you will see. Type 1 topoisomerase, they don't need ATP for the cleavage activity, that is the cutting activity, the break activity that is done in one of the strands. Whereas topoisomerase 2 cleave both of the DNA strands, it cuts both of the DNA strands. For this they don't require energy. Topoisomerase 2 is requiring energy only because for their conformational change in their structure during that process. So I have said that type 2 topoisomerase, they make double stranded breaks. Sorry for that. Okay, wait for a minute. So type 2 topoisomerase, they actually make double stranded breaks in the DNA, through which they pass a segment of a uncut DNA helix before receiving the break, and it requires energy because of its conformational change in the structure during their activity. Whereas type 1 topoisomerase, it makes single stranded breaks in the DNA that allows the uncut strand to be passed through the breaks before resealing the nick. Nick means the cut region. So type 1 topoisomerase make only a single strand break, and it allows the uncut DNA strand to pass through that break, and due to which it overcomes the supercoiling activity, it becomes normal coiling, and then it reseal the nick, it reseal that break. Type 1 topoisomerase completely don't require any ATP. Now both prokaryotes and eukaryotes have type 1 and type 2 topoisomerase, which are capable of removing the supercoils from the DNA. You will see that in eukaryotes also they have this topoisomerase. In prokaryotes they also have topoisomerase type 1 and type 2, which overcomes the supercoiling of DNA. Now I have given a little bit notes about the supercoiling of DNA, but before that, you see how topoisomerase 2 is overcoming the supercoiling of DNA. Suppose this is one extra coil that occurs in the DNA, and topoisomerase 2 tries to solve that supercoiling. So what it does, you can see that both the DNA strands are being cut down by the topoisomerase enzyme, topoisomerase type 2 enzyme, so that this helical portion, which is not cut down, it again it moves to uncoil the DNA, to uncoil the DNA helix structure, and then again that double strand is being resealed back. So you can see the top duplex. You can imagine, you can see the top one, and on the other side you can see the bottom one. Top one has to be removed that side. So for that they have cut both of the strands, and they allow that behind double helix to come forward. You can see they come forward, and again they reseal it behind. So this is the activity done by topoisomerase 2, right? And this is the mechanism of topoisomerase 1. It's very simple. You can see that here unwinding of the DNA. Both they can go for uncoiling or recoiling of the DNA. That means they undergo positive supercoiling as well as negative supercoiling also. So here negative supercoiling has been shown here. So you can see that one of the strand has been cut with the help of topoisomerase. Although topoisomerase here it is referring to a scissor, it is cutting one of the strands. You can see after cleaving it is saying pass strand through the break and ligate. That means the uncut that strand which has not been cut down, it passed through that break. It passed through that break, and after passing through that break, that that cut that strand which has been cleaved by the topoisomerase 1 is being resealed again, and thus we make that normal helical form of the DNA. So in this way your topoisomerase 1 and 2 carry out their function. Okay, now I have little bit said about the DNA supercoiling, okay, to understand the concept actually. So DNA supercoiling are of two types: positive supercoiling and negative supercoiling. Positive supercoiling of DNA occurs when the your right-handed double helix of the DNA is twisted even tighter. DNA is always a right-handed double helix that I have said. So if that right-handed double helical conformation or structure of the DNA is even twisted more, that means twisted in a right-handed fashion, it will be called as positive supercoiling, and due to positive supercoiling there will be a knot, git, there will be a knot formation on that position of the DNA. So as DNA is a right-handed double helix, what happened due to positive supercoiling? DNA is twisted, DNA is coiled more tighter. That is, they are becoming twisted in a right-handed fashion, and the helix begin to distort, and knot formation occurs. Whereas negative supercoiling, on the other hand, it involves twisting against the right-handed helical conformation. That means in negative supercoiling, the twisting or the coiling occurs in a left-handed fashion, and we know that DNA is a right-handed double helical structure, so it is coiling against the right-handed coiling nature of the DNA. So preferentially, the coiling will be now become loose, just like this. The coiling will become loose, just like this. And to overcome this problem, also topoisomerase come into function, right? So positive supercoiling is actually coiling with the right-handed conformation. They are twisting in a right-handed fashion, and DNA is also a right-handed double helical structure, so they are getting more and more coiled until the helix begin to distort and forms a knot. Whereas negative supercoiling is twisting or coiling against the helical conformation of the DNA. That means they are twisting in a opposite direction, in the left-handed fashion, which preferentially loosely coiled the DNA, and it straightens the DNA double helix at the twisting stress and knots into the DNA negative supercoils. Now prokaryotes have a special type of type 2 topoisomerase known as DNA gyrase, which can introduce negative supercoil. That means if there is positive supercoiling of DNA, topoisomerase 2, which is in prokaryotes, they have a special topoisomerase 2 called as DNA gyrase, it introduces the negative supercoil of DNA. DNA gyrase is responsible for negative supercoiling of chromosomes in prokaryotes. So they are responsible for negative supercoiling of chromosomes in prokaryotes, and this negative supercoiling will help the unwinding of the DNA helix, which stimulates many reactions of the DNA, including your initiation of transcription and DNA replication. So if you see from the transcription point of view or replication point of view, your DNA gyrase, as it introduced negative supercoiling in the chromosomes in prokaryotes, due to which the unwinding of DNA occurs. So it creates a platform where all the other enzymes will initiate the replication process or your transcription process. Like in prokaryotes, sigma factor is required for transcription. Okay. So now this is a diagram or figure which has been shown. So this is actually, it tries to show the coiling nature of the DNA. You see the bDNA. Normally we, when we draw the diagram of DNA structure, we usually draw the bDNA form. The left-handed DNA is observed on your Z form of DNA. The Z DNA is having the left-handed helical conformation. You can also see the overwound bDNA, the positive supercoiling. It is coiling with the helical conformation. That's why overwinding or winding of DNA occur, over coiling of DNA occur. And you can also see the underwound bDNA, where the coiling of the DNA, the undercoiling occurs against it, it coiling occurs on the left-handed conformation, and DNA is a right-handed double helical structure. So the coiling nature of DNA is now become loose. In this way, the DNA coiling occurs, supercoiling observed. Now this is a figure of trying to show that how the left-handed, it shows the topology of the DNA supercoiling actually. So you can see here, you can imagine the DNA double helix to be a two-stranded right-handed coiled rope. Okay. Now if one of the end of this rope is rotated in counterclockwise, means anti-clockwise, the strands begins to separate. Okay. Counterclockwise means moving in left-handed fashion. Okay. So here it is written actually. So then we will see the figure. So as it is saying that DNA double helix can be compared to a two-stranded right-handed coil rope. Now if one end of the rope is rotated in anti-clockwise direction or counterclockwise direction, the rope become overwound. Sorry, I have written, I have mistakenly showing the wrong line. So counterclockwise means anti-clockwise. So if one of the end of the rope is rotated in counterclockwise direction, the strand begin to separate, because it is moving against the helical conformation of the DNA, which leads to negative supercoiling. Now, if the rope is twisted in a clockwise direction, now clockwise direction means moving, rotating in a right-handed fashion. That means the DNA is also a right-handed double helical conformation structure, so it also get twisted more and more and becomes overwound, or you can say positive supercoiling. Okay. Now you can do the experiment by yourself by using a right-handed multi-stranded rope and try to twist in the left direction to get that positive negative supercoiling phenomenon, or you try to rotate in the right-handed fashion to get the positive supercoiling concept. So it has been shown here. The left-handed counterclockwise twisting is occur, which is analogous to the negative supercoil in the right-handed helix, such as your DNA. Okay, and this is the right-handed, which is the counterclockwise twist, which is analogous to the positive supercoil in a right-handed helix, such as bDNA. So when the DNA is twisted in a right-handed fashion, more coiling occurs, due to which it leads to positive supercoiling. So you can see if you compare the rope with DNA. So this is the circular nature of DNA. I have already said that prokaryotic DNA are circular. Most of the case, almostly they are circular. And this is the form when they are in relaxed. But when they undergo your positive supercoil, what happens? You can see, due to positive supercoil, the nature of the DNA, not formation occurs. Okay, right-handed fashion, in the left hand right-handed fashion, the positive supercoil is being observed, while here only one twist has been shown. If more twist occur, then not formation will be observed. Okay, while negative supercoil will lead to loosely unwinding of the DNA, because it is moving against the direction of the helical conformation of the DNA. Now, a special property which is done by the topoisomerase at the end of the replication in prokaryotes is catenation and decatenation. So, it is saying that topoisomerase can both catenate and decatenate the circular DNA molecules. Circular DNA molecules are said to be catenated if they are linked together like two rings of a chain. So suppose, as I have said that your the bacterial DNA is circular. Now, when this circular molecule DNA molecule has completed your replication process, the it becomes just like two rings attached together. So these circular DNA molecules are said to be catenated. Okay, just like this, I'm trying to show you the diagram. You can see the it will look like just this. So when replication has been occurred fully, then you can see the two new daughter DNA molecules containing one parental strand and one newly synthesized strand are being stuck together like this. This is called catenation. Now it has to be decatenated, right? For decatenation, topoisomerase 2 actually comes into play because it has to cut both of the strands of the DNA, and then to then it pass on that unwounded double helical strand so that they can really release by themselves, and then resealing occurs. Okay, mostly it is done by topoisomerase 2. Sometimes topoisomerase 1 can also play that role if if one of the strand has been cleaved. If one of the strand has been cleaved, at least one of the DNA molecules carries a nick or gap, then only your topoisomerase 1 can cleave that side, one of the strand, and overcomes that catenation process, release them, and again reseal them with the help of their ligase activity. So same thing has been written here, that that topoisomerase can do catenation and decatenation of the circular DNA molecules. Circular DNA molecules are said to be catenated if they are linked together just like two rings of a chain. And of these two activities, the ability of topoisomerase to decatenate the DNA is of special importance, because the DNA which has been prepared during the S phase of your cell cycle, not in prokaryotes undergoes the binary fission, budding process, so that type of process. So when DNA replication occurs, now they have to be distributed among the daughter cells. So normally catenated DNA molecules are normally observed when DNA replication is finished. Topoisomerase play an important role by unlinking that DNA molecules to allow them to separate into equally into two daughter cells for cell division process. So decatenation of two covalently closed circular DNA requires the passage of two DNA strands of one molecule through a double stranded break. And that double stranded break is actually done by the topoisomerase type 2. That's why decatenation explains that why topoisomerase type 2 are essential for cellular proteins. So you can see here that topoisomerase type 2, from the right, if you see, catenation occurs, okay, and it occurs during the time of replication. This thing occurs during the time of end of replication, end of replication. So during the end of replication, this type of thing is observed, and for this we have to cleave them. We have to cleave the two strands, both strands. Okay, I'm using different color, okay, red. So we have to cleave both the strands so that we can pass on this the uncut DNA helix to over to that cutted region, and then we will reseal them with ligase, and that is done by your topoisomerase type 2. And this now two DNA molecules can be separated equally among the daughter cells. The same thing topoisomerase type 1 can also do, but it is saying that however, if at least one of the two catenated DNA molecules have a nick or a gap, then only type 1 enzyme can also unlink the two DNA molecules. So, so you can see that if topoisomerase type 1 can also carry out this decatenation process if at least one of the two catenated DNA molecules have a nick or a gap. Obviously, here you can see there is a nick or a gap created here. One of the strands has been cleaved by some enzyme. So topoisomerase type 2 can carry out the function by cleaving at this site. Then it passes that uncut DNA double helix to that site, and again it reseals that nick. reseals that cutted region. Okay, now I will try to discuss with you about how the topoisomerase type 1 and type 2 carry out that function of cleaving and rejoining the DNA. They don't require energy. So how they do that? Let us see. So if I show you the figure, you can able to imagine. Then I will say in the notes. Okay, I will try to explain in the notes, but in the figure you can able to observe this phenomenon in a quite easy way. So here it has been shown the figure of your topoisomerase type 1, right? It has been shown the topoisomerase type 1. So you can see that both, whether it is topoisomerase type 1 or type 2, both topoisomerase type 1 and type 2 have a active, in their active site there is a tyrosine residue found. So you can see a tyrosine residue has been observed in the active site of the topoisomerase enzyme. So this is the active site they have shown, and in that active site there is a tyrosine amino acid residue. Now the DNA cleavage or DNA cutting occurs when your tyrosine residue is found in the active site of this topoisomerase will attack the backbone of the DNA. So you can see here, this is your DNA, this one is your DNA, and topoisomerase is, with the help of their functional group, it is trying to cleave, it is trying to cut this, the phosphodiester backbone of the, or you can say the phosphodiester bond of the backbone of the target DNA. Now this attack done by the tyrosine amino acid residue found in the active site of the topoisomerase to the DNA, this attack creates breaks in the DNA. So here the breaks occur is single. If it is topoisomerase type 2, then it will create double breaks by the activity of two tyrosine residues found in their active sites. But here we are showing the type 1 to make it more simpler. So here the attack done by your tyrosine amino acid residue found on the active site of the topoisomerase type 1 to the backbone of the target DNA, this attack creates a break. So a break has been created. You can see in one of the break, the phosphate group that is being formed at one end of the DNA strand has been attached with the topoisomerase of the active site of the enzyme. So this attack generates break in the DNA. The topoisomerase is now covalently attached, it is now covalently joined to one of the broken DNA ends via the phosphotyrosine linkage. So this is a phosphotyrosine linkage. This linkage is now called as phosphotyrosine tyrosine linkage. So phosphotyrosine linkage occurs. Okay. Another strand is now free. It has three prime OH group at one end. It will catalyze the next reaction you will see later on. So the other end DNA has three prime OH ends, and this end is held tightly by the enzyme. So this end is not loose actually. This end is held also tightly by the same topoisomerase enzyme, although it has not been shown. It has shown only on this much, but this enzyme has also influence on this part also. It holds tightly with the help of phosphotyrosine linkage on this site on one of the broken ends by help of phosphotyrosine linkage. On the other end, the other end also have a 3 prime OH group terminates that broken DNA fragment which has been attached with the topoisomerase enzyme non-covalently, obviously. Now this phosphotyrosine linkage is actually conserving this energy. So this phosphotyrosine linkage is conserving the energy, and the energy which was being when it was cleaved, when we cleave the phosphodiester bond, that energy is being conserved by this linkage, and therefore the DNA can be resealed simply by reversing the original reaction. That is, now the OH group, the 3' OH group will, from the broken DNA end, will attack that phosphotyrosine bond. So you can see the OH group is attacking to this phosphotyrosine bond and rejoining the DNA phosphodiester bond. Now what's the deal then? Why do this? It is breaking first and then is resoiling. By that time it moves that uncut DNA strand to this nick. Okay, so this reaction rejoins the strand, and by that time, before rejoining of the strand, in this cut position, the uncoiled, the uncut strand is passed through this site, and in this way we can able to overcome the supercoiling nature of the DNA. Okay, and it will now catalyze the another reaction cycle. So the same thing you can see if I show you the series of steps. You can see, you can able to observe that how topoisomerase 1 carry out this function. Now in the B figure that is shown here, it is actually the phosphotyrosine linkage which has been shown, that how phosphotyrosine linkage is being carried out by the enzyme, the structure of the phosphotyrosine linkage. Now in case of type 2 topoisomerase, they are dimer. That means within their active site they have two active sites. They have two active sites. Sorry, not two active sites. They have two tyrosine residues on their active sites, and the same process they do. If one tyrosine residue on the active site of type 1 topoisomerase can cause one strand cut, then type 2 obviously they have two tyrosine residues on their active site. They will create double stranded cut. Okay, so this is the nature of your type 1 and type 2 topoisomerase. So just go through the notes now. So it is saying that topoisomerase have a tyrosine residue in their active site. DNA cleavage occurs when tyrosine residue in the active site of the topoisomerase. This tyrosine residue will attack the phosphodiester bond in the backbone of the target DNA. Now, due to this attack, it caused break in the DNA, obviously. So the topoisomer is now covalently joined to one of the broken ends of the DNA with the help of phosphotyrosine linkage, while the other broken DNA end has been ended with a free OH group. Now this end is also held tightly by the same enzyme. Now this phosphotyrosine linkage is conserving that energy. It actually conserves that energy which has been formed when we are breaking that phosphodiester bond initially. So the DNA can be resealed simply by reversing the original reaction, that is the OH group from the broken DNA end of the phosphotyrosine bond will be attacking. So the broken DNA end which has the OH group, it will attack the phosphotyrosine linkage and reforming the DNA phosphodiester bond. This reaction will rejoin the DNA strand and releases the topoisomerase, which can then go on to catalyze another reaction cycle. Okay, so this was all about your topoisomerase function. Well, this has been shown that about the type 1 topoisomerase, that how it carries out the function. Try to show with the help ribbon shape model. Well, the twisting things that you can see here, the twisted things that you are seeing here, it is the alpha helix conformation, while as the line figure is your beta pleated sheets. The alpha helix, beta pleated sheets are observed in the secondary structure of the proteins. So this is actually your structure of the secondary structure showing the topoisomerase, right? And these are the subunits of the topoisomerase enzyme. So and these black lines are indicating the DNA. So you can see that how cleaving is done by the how cleaving, or you can say cut, done by the topoisomerase enzyme, and how that unwinded uncutted strand is passed through that nick, and then after passing they are resealing that nick, and we are releasing the DNA. So in this way your topoisomerase carry out the function. Okay, so that's all for today's lecture. I hope you have understood this part of topoisomerase. In the next lecture, I will try to discuss about the theta mode of replication of bacterial DNA, and also the rolling circle mode of DNA replication. If you have any problems regarding this lecture, if you have any queries, you can ask in the WhatsApp group. Okay.